Dear learners, Namaskar. I'm Dr. Nutan Prakash Vishwakarma. And in this video, I'm going to demonstrate how to perform the local pairwise sequence alignment in the two protein sequences. If you have the DNA sequences, you can perform by using the same steps. So a lot of softwares and servers are available. Here I'm demonstrating by one of the very important server that is known as EMBOSS. So just like in the EMBOSS, there was the one domain, that was a needle that we have used for global alignment. Here I am going to use another tool that is known as water, which is available with the EMBOSS for the protein sequence alignment, especially the local sequence alignment. So for simplicity, what I did here, I have the two protein sequences, and in these two protein sequences, I want to know that how much percentage of the similarity both are sharing locally. So here a protein sequence number one, here I do have the protein sequence number two, and I told you once there is the huge difference in the length of the two protein sequences or DNA sequences, our two sequences are irrelevant. In that case, we can perform the local sequence alignment and local pairwise sequence alignment. So let's move ahead how to perform. So simply just click on Google the EMBOSS. Here you will get the link of EMBOSS program. In the EMBOSS program, there is a lot of softwares are available. So here I use for the local alignment, here use the water algorithm. As you know, for the local sequence alignment, we use the Smith and Waterman algorithm. So water is actually name have been taken from the Smith-Waterman algorithm. So simply just like if you have the protein sequences, you can keep select as a protein. Otherwise, if you have the DNA sequences, you have to DNA to for the alignment. Then you can click on the DNA. So here I have two protein sequences. So simply I selected the protein. And here in this case, I have two protein sequences. So protein sequence number one I paste in the first box, and protein number 2 I paste on the second box. And by using the default parameters, I move ahead for the sequence alignment, that is the pairwise local sequence alignment. So it depends on the internet speed, maybe within 1 or 2 minutes you can get the result. Here you can see that aligned sequences were 2. The name of the sequence was 1 and 2, that just for demonstration point of view I put it the name as 1 and 2. And here the gap opening penalty was 10 and extension penalty 0.5 was used. You can change it also if required. The length of the sequence which was aligned, that is 240, although the length of the first sequence was more, and the second length was the length of the second sequence was less. So here you can see that the starting from the 61, all right, so starting from the 61 amino acid up to 300 amino acid of the first sequence is matching with the 235 bases of your second sequence. So starting from 1 to 60, there was no match. And maybe after 300, so 393 was the length of the first sequence. So after 300, the sequence is not matching with the second sequence. And here you can see this is the reason, this is the total region which is absent in the sequence number 2. So overall, locally, your sequence number 1 and 2 having the 97.9% identity. Since there is the only gap here, 100% identity is there, only due to few gaps you got the 97.9% identity, and 2.1% here is the gap. So that's how once you have the two protein sequences with you, or you have two DNA sequences, by using the EMBOSS water, you can easily perform the local sequence alignment among two sequences. So I hope this tutorial is going to help you for the pairwise sequence alignment. So see you soon in next video. Till then, thank you.